Hello everyone, my name is Jiawen Yang. I'm a first-year PhD student from the Department of Cancer Biology at the University of Arizona. Today I'm going to present my poster named The Network Analysis Reviews Transcriptional Regulation Driving Neuroendocrine Tumors. To begin with, I will give a brief introduction of neuroendocrine tumors. Neuroendocrine tumors are a heterogeneous group of malignancies with both neural and endocrine cells properties. They can rise in multiple tissues and originate from either mature neuroendocrine cells or from non-neuroendocrine cells. They can evolve to acquire neuroendocrine features, and all the neuroendocrine tumors exhibit similar morphology and often express common markers, which suggest a shared transdifferentiation process drives their development. While Merkel cell carcinoma is a rare but highly malignant neuroendocrine tumor, it expresses the same neuroendocrine markers that are expressed in a Merkel cell. One-third of patients present at primary diagnosis with local regional metastasis. That's also why this tumor has high mortality rate at about one-third. Different from other neuroendocrine tumors, the carcinogenesis of Merkel cell carcinoma is associated with the presence of clonally integrated Merkel cell polyomavirus, so that the mechanism of which can be studied in vitro. The viral etiology of Merkel cell carcinoma therefore provides us with a unique opportunity to investigate the regulatory circuits driving neuroendocrine tumor formation and metastasis. To study the transcriptional regulation in the process of carcinogenesis, we used the normal human long fibroblast cell line IMR90 as the parental cell and transduced with doxycycline-inducible vectors carrying Merkel cell polyomavirus oncogenes, small t, large t, antigens, and ER, which stands for early region containing both ST and LT, or GFP sequence. We track the influence of the oncogenes over time through RNA-seq profiling. Using the dynamic RNA-seq data, we computed the differential expression of genes and applied a model-based clustering to genes that perturbed from different oncogenic conditions. Clusters were enriched to GO terms to provide potential phenotypical changes under different conditions. To gain an insight into the mechanisms of the activation of neuroendocrine markers and the programs in the process of IMR90 cells to Merkel cell carcinoma transition, we used our samples to infer a cell line-specific transcriptional network using the algorithm PANDA to detect the changes in the activities of transcription factors. We also applied algorithm alpaca to identify the genes and transcription factors that potentially drive the IMR90 cells to Merkel cell carcinoma transition. As we can see from the heat map, comparing each oncogene expression cell line against the GFP cells at the same time point, genes are differentially expressed in ST, LT, and ER groups. Clustering the genes reveals that LT and ER upregulate the expression of cell cycle-related genes. Both ST and LT alter the expression of neuroendocrine markers, developmental regulators, cell motility genes, and EMT pathway members. Next, we applied model-based clustering to the genes perturbed by ST, LT, and ER, and we find out both ST and LT perturbed genes were enriched for GO terms related to cell migration. We observed that ST activates expression of genes that are involved in neural cell migration and motility, including DPYSL3 and PTGS2, which shows in the heat map in panel A, whereas LT significantly upregulates Daphne family genes and ECM-related genes, which are known to promote metastasis in cancer cells. ST and LT both increase the expression of Twist and alter expression of TGF-beta pathway members. We observed that LT upregulates SMAD145 and ST upregulates SMAD2 and 9, whereas the repressors SMAD6 and 7 are both decreased in ST and LT cells, as it shows on panel B. TGF-beta pathway members are differentially expressed by Merkel cell polymervirus oncogenes. Other major signaling factors like FZD9 are activated only transiently. In panel C, we observe that the ER caused increased expression of neuroendocrine and Merkel cell carcinoma markers, such as ENO2, NEFM, and CN1. And then we applied alpaca to compare the ST, LT, and ER networks with GFP network. The nodes are colored by membership in differential modules. Network is annotated with representative enriched goal terms in each module. The nodes with annotations are leading-edge neurodevelopment genes. We discovered that ST, LT, and ER activates modules differently. In ST, the top-ranked genes in the modules are enriched in goal terms related to regulation of RNA metabolic process or aromatic compounds biosynthesis process, whereas in LT and ER group, the top-ranked genes in the modules are enriched in goal terms related to neuron recognition or positive regulation of neurogenesis. Networks from the three groups suggest that neurodevelopment is mostly contributed by LT. We find that LT activates a module of genes regulated by transcription factor ATON1, which is the key regulator of neuron formation that is also involved in the development of Merkel cells. Recently, it was found that LT activates ATON1 to induce the neuroendocrine cells morphology. In conclusion, our data suggests that ST and LT perturbed complementary aspects of cell motility and developmental pathways. And our current results shows that LT activates a module of genes that contribute to promote the neuroendocrine tumor transition. And also, further functional and mechanistic analysis are needed to address the changes in the regulatory network behind the carcinogenesis process. Overall, our study systematically investigates and quantifies the regulatory circuits driving neuroendocrine tumor formation and metastasis, which could potentially shed the light to the development of targeted therapies for both localized and metastatic neuroendocrine tumors. That's all for my presentation today. Thanks for your attention.